Good morning and welcome everybody. My name is Kyle Miller, and today what I'm going to do is give a lecture about the essential cell biology for understanding how neurons grow. In the last class we sort of introduced the idea about actin, and today we want to talk about how actin is regulated in the context of cells. And as just a bit of context, so this was originally based on a lecture for MMG 409 eukaryotic cell biology, and I've just slightly updated it for you guys. So, all right. So in the last lecture, we had introduced the idea of actin assembly, and one of the things that we pointed out was that the rates of assembly are different on the plus and the minus ends of actin filaments. And what we're looking at here are actin filaments where the subunits have been fluorescently labeled, and we can visualize their elongation. And which of these two ends would be the plus end here? So on the right side, and indeed, so the plus end grows a lot faster than the minus end. And when we talked about the kinetics of this, we can understand that in the context that the rate of assembly depends on the concentration of soluble actin. And when you look at the sort of kinetics, what you'll sort of find is that for a given concentration of actin, rates of assembly are about 10 times faster on the plus end than the minus end. So assembly depends on the concentration of free actin, and then at that same time, there can also be disassembly from the filament, and of course that's going to be independent of the concentration of soluble actin, because this is all happening in the filament. If you put this, if you have an actin concentration that is at the critical concentration for an actin filament, and as just a reminder, the critical concentration is the steady state concentration where the rates of assembly and disassembly are balanced. So if the soluble actin is at the critical concentration, it is going to, the critical concentration for the solution is going to be between the critical concentrations for the plus end and the minus end of the actin filament. And when you bring these sort of ideas together, what that means is that you get assembly on the plus end of the filament because it requires a lower concentration of G-actin for assembly to occur. And you're going to get disassembly on the minus end of the actin filament. So when you look at in cells or at actin filaments where the G-actin is at the critical concentration, treadmilling occurs. And by that, what that means is we have a little actin filament at the top. We've got the plus end over here, the minus end over here. And because this filament is, the G-actin's at the right concentration, there's going to be assembly on the plus end, or actually a better way to start is actin is going to disassemble from the minus end of the actin filament, and then there's going to be assembly on the plus end of the actin filament. And if we just mark one pair of G-actin, or rather the actin subunits here, we can see that they would stay in place while the filament of it itself would appear to move through treadmilling. And what's really moving is the soluble actin is being moved to the front to build new actin filaments. And this is just a little movie that I had shown last time, and it sort of summarizes some of the big points. So during actin assembly, you get nucleation, which is a relatively rare event because it depends on multiple G-actins coming together at the same time. Initially, there's going to be an elongation phase where the filaments are growing longer on both ends. As that's happening, the concentration of G-actin is dropping. At steady state, where the critical concentration has been reached, there can be assembly on both the plus and the minus ends. But I think one of the shortcomings of this figure is it doesn't show this treadmilling, or this movie rather, it doesn't show the treadmilling, but it does capture a lot of ideas about actin filaments. Now, one of the key things is that cells need to regulate where actin assembles, and there are a lot of different proteins that control actin dynamics. And the next thing I want to do is start to talk about some of those proteins. And I'll just pause for a second. Everybody's good with critical concentration, steady state, those sorts of things, so, okay. All right. When we start talking about actin in the context of the cell, there are a couple of different proteins that are critical for regulating actin assembly and disassembly. So here we have up top an actin filament. The plus end is over here, the minus over there. You can see that when G-actin assembles into an actin filament, initially ATP is bound, and over time that ATP gets hydrolyzed to ADP plus PI. The inorganic phosphate stays in the actin filament though, and then eventually that phosphate is released and we're just left with actin and ADP. So at the critical concentration, there is going to be disassembly at the minus end, but it also turns out that when cells need to get rid of actin, they have proteins that are dedicated to that function, and one such protein is cofilin. And what cofilin does, so cofilin is these sort of smaller red dots. It binds to the actin filaments and gives it a little bit of a twist, a little bit of a torque, and that helps break these actin filaments apart. And then when you have a bunch of short actin filaments, you can get rapid disassembly because you have a lot of minus ends that the actin can fall off of. When that actin gets converted to the soluble state, it then needs to exchange the ADP for ATP. And there is a protein called profilin. It promotes filament formation. And what it does is it binds to the G-actin. So we've got the G-actin, you can kind of think of it like a hand. There's a cleft where the ATP goes in. But the profilin binds to the backside of the actin and it opens it up. And the profilin facilitates the exchange of ADP for ATP. And now we have a fresh G-actin with ATP bound. One little quirk of actin in cells is the concentration of actin in cells is about 100 times higher than what the cellular kinetics of actin would suggest. And part of the explanation is there's another protein called thymosin beta 4 that buffers actin. So what thymosin beta 4 is doing is it's binding to G-actin and it's blocking its ability to form filaments. And that stores a huge amount of G-actin in the cell that's ready to be used when needed, but otherwise will not assemble into actin filaments. And I'll go into more detail about these proteins as we talk here. So this goes through the profilin cycle. So we've got profilin here. So profilin binds the G-actin, opens it up, and essentially I've kind of talked through this. One thing is that profilin binds other proteins that are rich in prolines. So this is a specific type of amino acid found in proteins. And the sort of key here is it allows profilin to be concentrated in regions where actin assembly is needed. So profilin promotes actin assembly, and it's also localized where actin assembly is needed, much as we might expect. In terms of the thymosin beta 4 cycle, it turns out that only about half of total actin is soluble in vivo. The rest is in filaments, but it turns out that actin concentration is really high in the cell. So it's about 100 to 400 micromolar. And if we remember back to those numbers that I was throwing up from the in vitro biochemistry, the critical concentration of pure actin is about 0.2 micromolar. So it's about 200 times lower. And what that sort of indicates is that if you just look at the in vitro biochemistry, you would expect that all of the actin in the cell would be assembled in filaments. But when you look at the cells, it's not, and it turns out that thymosin beta 4 is the protein that explains that. And the way that it does that is it binds to ATP G-actin and it blocks its assembly onto the plus ends of actin filaments. And I mentioned that actin is at a really high concentration in the cell. Thymosin beta 4 is also at a really high concentration in the cell. So you have this protein that's blocking actin activity at a super high concentration, and it acts as a buffer. And if you think back to your chemistry class, the way that buffers work in, say, pH, and actually I'll just pause for a second. Do you guys recall how buffers work, like when you're doing pH problems and stuff? Would somebody want to jump in and briefly explain that?
Okay, so when you have a buffer, you have a solution with an acid and its conjugate base, or a base and its conjugate acid. And what happens is it resists changes in pH when a small amount of a counter ion is added to the solution.
Yes, exactly. So the thing that buffers do in pH is they prevent large changes in pH with small changes in the concentration of an acid or a base. Thymosin beta 4 is doing the same thing. So what happens is that as actin is being made, some of it gets pooled in with the thymosin beta 4, and it prevents the free actin concentration from getting really high. As actin goes into filaments, that lowers the concentration of G-actin in the solution, and then what that means is that the probability of G-actin being released from thymosin beta 4 increases, and then the concentration of G-actin increases. I don't know if I explained that smoothly, but you follow what I'm saying? So yeah, essentially the thymosin beta 4 is locking up all of the actin. If actin concentration goes down, then the thymosin beta 4 releases G-actin to boost the free concentration back up. And it really is very much like the pH problems that you learned in chemistry about buffering and whatnot. Okay. So, and then in terms of the cofilin cycle, once again, so cofilin's over here. It binds between sets of monomers. It induces a little twist. So when I was talking about torque, there's a force, and it breaks it down. And the big idea is that this creates a lot more myosin. So, all right. There's a couple of other proteins that are involved that we need to talk about. At some point, sometimes in cells, it's really important that actin assembly is completely blocked. So if you think about it, in your muscles are mostly actin and skeleton myosin 2, or skeletal myosin 2. And in your muscles, certainly when they're growing, there needs to be actin assembly. As the actin gets damaged and whatnot, it needs to be replaced. When your muscles grow, you're actually assembling new actin filaments. And there's a protein called CAP-Z, so it's a cap that binds to the plus end of actin filaments, and it blocks its assembly. And then likewise, there are other proteins, tropomodulin in particular, binds to the myosins of actin filaments. So there's proteins that are binding to the G-actin, so profilin, thymosin beta 4. There's proteins that are binding to the F-actin, the actin filaments, CAP-C, and tropomodulin. And once again, these proteins are controlling actin assembly. Yeah. All right. So once again, it turns out that CAP-C has a very high affinity for binding to the ends of actin filaments. So it's on the order of 0.1 nanomolar. And if we recall, so we've got nanomolar, micromolar, millimolar. So this is about 1 × 10⁻⁻⁻ molar is the binding affinity. So it's a very high affinity binding. And one question is, you would expect that CAP-C is going to completely block all actin assembly, once again, just based on the biochemistry, and it kind of leads to the question of how actin filaments are able to grow at all in cells. And one of the keys is that on the plasma membrane, there's these lipids, we would just call this PIP2, so, and it's found, there's lipids, PIP2 in particular, found at the plasma membrane. And what happens is PIP2 binds to CAP-C and inhibits it. And if we think back to the last lecture, one of the big points that I had made is that actin filaments are usually found on the outside of cells, right against the plasma membrane. And one of the mechanisms that allows this to happen is CAP-C. So CAP-C is going to be at a really high concentration at the inside of the cell. It's going to be active. It's going to be blocking actin assembly. And it's only right at the plasma membrane where CAP-C is being inhibited that actin filament assembly can happen at all. And yeah, so this is allowing actin assembling right at the cell membrane. And then once again, with the tropomodulin, so the tropomodulin is binding to the minus end of the actin filament. And one place that this is also found, I'm pretty sure tropomodulin is in muscles, we'll see in a second here. But yeah, it is. So red blood cells and in muscles. So if you think about it, like your red blood cells need to be able to resist forces. So as blood cells move through your body, there's a lot of pressure differentials that can tear blood cells apart. And in fact, in sickle cell anemia, one of the problems is that the blood cells lose their integrity because these forces are there. And it turns out that tropomodulin sits on the actin filaments next to the plasma membrane, and it helps keep the cells intact. So these actin filaments are useful for cells resisting forces and keeping themselves together. And tropomodulin plays a role there. Okay, so with that, that's kind of the foundation of controlling actin filaments. And the next thing I want to do is talk a little bit more about the cell biology. So taking a step out, we started with the in vitro stuff. We've now introduced some proteins. We're now going to look at whole cells and talk about how these things fit together as a whole. So this is a picture of a growth cone from an aplysia neuron. And for those that might not know, so aplysia is a type of sea slug. They're about as big as your hand. They're huge. They're actually really beautiful animals. So at least the ones I've seen are black and they've got like some bright coloration and stuff, and they swim around. And I'm sure if you've seen nature documentaries, maybe you've seen pictures of sea slugs. The thing that makes them special for cell biology is that the neurons in aplysia are gigantic. So if we think of our common sort of like axon to be about 1 micron across, the axon of aplysia is about 10 microns across. So the diameter is 10-fold larger. And if you think of an area of a circle, you can see that that's going to mean that there's a huge volume of material in the axons of aplysia neurons. And because they're so big, that's one of the reasons that people do cell biology on them, because you can do a lot of really neat experiments with them. In any case, so here's the growth cone of an aplysia neuron. This would be the leading edge of the cell, and we would call this the peripheral domain of the growth cone. So it's out on the edge, and the plasma membrane would be found right here. So if we look on the center here, you can see there's lots of sort of little bright spots. Those could be organelles or whatnot. But probably more importantly, these lines here, at least the thinner ones, are going to be individual microtubules that are in the central domain of the growth cone. So in the growth cone, you have a central domain full of microtubules and organelles, and there's a peripheral domain that's rich in actin, and it's right against the plasma membrane of the cell. In addition, I won't talk so much about neurofilaments, but there are also neurofilaments that are found in the C domain, and if we did some immunocytochemistry or whatnot, we would be able to see at least a few in the early stages of development. Okay, I guess the one final thing I'll point out, so if you look at the structure of the growth cone, you'll see that there's these sort of little sharp extensions that go out. They kind of look like fingers. These are the filopodia in the growth cone. Filopodia, everything out here is actin, so these filopodia are built from actin filaments. And then the lamellipodial veil is right here, and that's going to be a cross-linked meshwork of actin filaments. And we'll talk about the proteins that build up these different structures in a bit. Yep. So, oh, yeah, this is actually kind of nice. One thing that's really cool about this figure, so we've got what's called a quick freeze deep etch EM of a growth cone, and it's paired with immunocytochemistry. People are familiar with quick freeze deep etch electron microscopy? Okay, the way that it generally works is what you do is you take a cell and you really rapidly freeze it. So typically people would like to use liquid helium to very rapidly freeze the cells. When cells freeze very rapidly, large ice crystals don't form and the cells don't get destroyed. Once it's been frozen, the water gets sublimated, the ice gets sublimated, and then there's just kind of a structure of proteins that are left, and then platinum is evaporated, and it then condenses on the different proteins that make up the growth cone, and you then take this little sheet of platinum and you put it in your electron microscope, and you can get beautiful images like this. Or you can actually, in this particular case, I think this is just viewed directly. So what is actually also kind of cool is this is also paired with immunocytochemistry, where the microtubules have been labeled up in blue and the actin filaments are in red. And this just really reinforces the idea that the microtubules are in the center, in the C domain, and the actin filaments are in the periphery, in the P domain. So we've talked about profilin, we talked about cofilin. One of the proteins that is important for modulating and regulating actin assembly are the formins. So they are ubiquitous, which means they're everywhere. And they have these two domains. They have a forkhead, I think, is it forkhead or formin? So they have an FH1 and FH2 domain that are donut shaped. And once again, in this particular case, the F1 domain is rich in prolines. And as I mentioned, profilin binds prolines. So one of the keys to localize profilin at the membrane is through formins. So that's the kind of link between these two things. And as I'll show you, these two things work together, and they have a stepping motion that assists actin assembly. And this is a little movie that just shows that. So here is our actin filament. We've got our plus end up here. So the formins are going to be sitting on the plus end, and they sort of walk back and forth, and they help that G actin bind to the F actin selectively at the plus end. And one of the ways that they do this is that this formin has a domain that protects the plus end from cap C. So you can kind of see how these things are happening. Actin assembly is going to be at the membrane. Cap C is going to be everywhere. Cap C is going to be blocking assembly. And it's only at the membrane where profilin and formins are working together, localizing the actin filaments at the plus end, turning off cap C, protecting the end from cap C, allowing actin assembly to occur. And a really big theme in cell biology, and one thing that makes all of these processes really complex is that there's usually multiple mechanisms that are tightly regulating how things assemble and disassemble. And certainly the biochemistry is essential for working out all of the sort of like different like parts and things, but you need to start looking at it as a whole and putting it all together to really understand how cells work. So, and that's the essential balance in all of science, this sort of top-down versus bottom-up approach. And it's useful at different times to flip back and forth between these two. Okay, so how could we test the role of formins in actin polymerization? Go ahead.
Do two experiments, one with extra formin and one without formin?
Exactly, yeah. So the heart of any experiment is you set up an assay, you've got your control, you've got your experimental group, and you do the experiment. So in this case, this was the movie that I had just shown at the beginning. And what would you expect occurs to the rate of actin polymerization when formin is added to the solution here? Just naively based on what we've just talked about. So if you've got actin filaments, nothing else, you just add formin to the solution, what do you think is going to happen? Go ahead.
It's going to help actin to bind.
You definitely, and that's yeah, exactly. So that's kind of what the expectation is. And right here are the two movies. So this is just actin itself, and then this is actin plus formin. So watch the movie and tell me what you see. Go for it, yeah.
The actin plus formin experiment has a lot more of the F-actin actually growing.
That is definitely a possibility. I hadn't really looked at that, but it just could be that when they were taking these movies, there was a certain part of the cover slip with a lot of actin filaments and another region with fewer actin filaments. Go ahead, Chloe.
Actin alone seems to be growing more rapidly, or
In-
moving at least more rapidly.
Yeah, yes, yeah, go ahead.
I think the minus side of the one with formin is at one place, it's not moving. And
It looks very static, yes, yes. So the big takeaway I was trying to get at with this is it's certainly not dramatically faster. So just in terms of this sort of initial hypothesis that, okay, so formin binds to the plus end of actin filaments, hey, maybe it's making the actin filaments grow faster. So that's not really supported. And instead, if anything, it kind of looks like it might be a bit slowed. Okay. Now, which actin-associated protein that we've discussed so far is also involved in actin assembly? Profilin, indeed. And what do you think would happen if profilin alone was being added to actin filaments?
it will make it grow faster.
You would think it'd make, exactly. Okay. So that's this experiment. Does that support your hypothesis? Indeed. So it's a bit puzzling and stuff. Okay. And, a lot of times when you're doing science, we've got a protein that binds to the plus end of actin filaments. We knock it out in cells and we see there's less actin filaments. We're absolutely sure that it's going to make actin filaments grow faster. We do the hardcore sort of like biochemistry, cell biology, and we see that it doesn't. And what this is telling us is we don't understand how this works. It's got to be more complicated. There's got to be something else going on here. And now the question is, if you would mix the combination of profilin, actin, and formins, what would you expect to happen?
I mean, based on the videos that we've seen, we would have F-actin growing at a very controlled rate, very slow.
Exactly.
Probably wouldn't degrade.
Exactly. Yeah. So you do one experiment, the things grow a little bit more slowly. You add a different compound, they grow a little bit more slowly. If you add both of them, you would perhaps expect an additive effect where they're going to grow even more slowly.
Okay.
Yeah, no. But just thinking through these results is really helpful. And when you're dealing with problems, not just in science, but everything, you do one thing, you do another thing, you mix them together, and then something at least a little bit unexpected happens. And you want to understand how that works. And I'll just sort of lay it out for you. So up here, we have the formins. So this protein up here, so this is the FH1 domain, FH2 domain that we just talked about that are stepping back and forth. We can see that profilin links the G-actin with the formins here. So the sort of trick that makes this all work is when you have these two things together, you have a functional machine that then can interact with the plus ends of actin filaments and drive really rapid assembly. So yet another idea is one way that cells ensure that actin assembly only occurs where it's needed is you need a combination of proteins to come together at the right place in the right time in order to nucleate actin assembly. And this is, yeah, it's a nice little sort of example of real experiments and stuff.
Almost like an assembly line.
Yeah, it is, in fact, it very much is. So, yeah, go ahead.
So in an experiment, how do you know whether you've taken the reductionist view so much that you've lost an emergent problem?
It's hard to say. If your life is full of confusion, perhaps you might want to start thinking about bigger picture sort of things. That might be the case.
Yeah.
I will say, the strength, ultimately, you need both approaches. So you need the careful, like, what is the binding affinity of this protein for this when they're absolutely alone, and for this and this when they're absolutely alone. So you build up a foundation of knowledge, and then you start to work higher. Another way, and in fact a lot of what we do in the lab, is we look at whole cells and we ask how neurons grow. And what we're doing is we're taking more of a top-down approach of, like, we want to understand how axons grow, we understand at least a little bit of the physics and stuff, and then we're going to disrupt different aspects of signaling pathways that control actin assembly and ask how that changes axonal outgrowth. So, okay. All right. So this is a nicer sort of fuller view. There are a class of proteins called the Rho family GTPases. And Rho family GTPases break down GTP. And when we were talking honors biology, when we were talking about GTPases and whatnot, this is exactly what we were talking about. So these are the proteins. They play a lot of roles in controlling different things. And in this particular case, there is you can have Rho itself. So Rho is a protein that is found in the Rho family GTPases, which will seem very confusing. But Rho is sort of, my understanding, it was discovered first, and then that became the sort of Rho family GTPases, and then later on they discovered things like RAC, CDC42, RIF, and a lot of these other proteins that all are related to Rho. So when you hear Rho family GTPases, you're thinking of the big class of proteins. If you hear Rho, or in particular Rho A, we're talking about a specific protein, and that can be confusing sometimes for people. All right, so we've got our plasma membrane up here, and this little sort of curly line, that's going to be a hydrophobic region that's going to allow this to bind to the plasma membrane. When GTP is bound with this, it's going to bind to profilin, and it's going to open up the profilin, and it's going to allow these different domains to interact with the actin filaments and the G-actin. And this all really just sort of links actin assembly to the plasma membrane. So the regulator of it is attached to the membrane. It activates this formin, and then these other proteins can work along with it to build activate ones. Okay. And just as a reminder for the Rho family GTPases, the way they work is we'll start off with Rho bound to GDP. There's going to be a guanine exchange factor that's going to exchange that GDP for a GTP. That is being activated by a receptor on the membrane. So when I was showing that movie of leukocyte chasing after a bacterium, it was sensing the outside world. It was driving actin assembly where it needed to happen in order to move the cell towards the bacterium. And what you can imagine is that there are receptors on cells that are binding signals, and when they get these signals, they activate GEFs. These GEFs can activate Rho, and then Rho turns on effector proteins like formins and profilin to drive actin assembly where it's needed. When that signal disappears, let's say the bacteria moves someplace else, the cell needs to reorient. There's going to be GTPase activating proteins called GAPs that are going to convert that GTP to the GDP state, turning off the protein. And then in addition, there are other proteins, so there are inhibitory proteins that can also bind to Rho itself and just turn it off. So there's a couple of different mechanisms for regulating this. But the big sort of picture is signals from the outside world activate GEFs. GEFs turn on Rho family GTPases, and then that turns on effector proteins, controlling where cytoskeletal elements are formed and disassembled. And it turns out that different Rho family GTPases lead to different actin structures in cells. So for example, RIF and CDC42 in particular are associated with filopodia, those finger-like extensions. So they have a really unique structure, and it turns out there's a unique set of Rho family GTPases that create them. RAC drives lamellipodial formation, so that's over most of the P domain of the growth cone. And then Rho itself activates stress fiber formation and contraction. So in neurons, there aren't stress fibers per se, but you do get bundles of actin filaments and whatnot. And Rho is actually really important for axonal elongation. It's one of the few targets that seems effective in promoting regeneration of neurons in the body. So Rho is really important for actin filament assembly, but Rho usually makes really static actin structures. And if you think about axonal elongation being a dynamic process, if you make the actin more static, it inhibits it. It's a lot more complex than that, but I'll just, go on. Go ahead.
I know this is a very simplistic view of it, but understanding that all four of these proteins, or like RIF, CDC, RAC, and Rho, they're all bound to the plasma membrane. How do they, are they different lengths? Is that how they're able to form into different sections of the filament that we're seeing in the photos below?
Yeah, I think what more typically happens is that these proteins would be more concentrated or more active
in the tips of those.
At the tips of the filopodia, and there's a lot more going on here. Big sort of picture idea is there's different Rho family GTPases that create different actin structures. and the details get super complex very rapidly. One of the big, actually, and one of the things that I'm working on right now is this paper about how neurons evolved. And one of the themes that seems to be emerging from that is it looks like a lot of the evolution is happening at the level of these row family GTPases that control where cytoskeletal assembly and motor activity happens. So if you want to make cells with different shapes, you need to turn on different motors and assemble the cytoskeleton in different ways. And because these control those properties, my understanding of the problem that's sort of emerging is that my guess is that there's mutations up at these high level aspects of the signaling pathway that cause cells to get their unique shapes. And neurons have a very unique morphology, and that seems like a good explanation, the best explanation that I have right now. So.
Are these all G proteins?
Yes, yeah. Yes, definitely. So the question was, are these all G proteins? So G proteins bind to GTP, and that's why they're called G proteins. So indeed they are, yes. So, okay. All right. So the next thing I want to talk about is, so we've got this lamellipodial structure here, the P domain of the growth cone, and I want to talk in particular how the mesh of the lamellipodium is formed. And for here, what we've done is we're really zooming in at the very leading edge of the cell. So if we were to go here and we were to take a small region about like this and zoom in, this is what we would see. And up here at the top is just the substrate. And these lines here, these are the individual actin filaments in the lamellipodia. And if you even zoom in closer, you can see that in fact, if you kind of look at a global view, it just looks like a big mess. Like there's filaments everywhere. But if you really zoom in, it actually turns out that the actin filaments are branched. And you can sort of see, so this would be the base of an actin filament. Here's a new actin filament that's branching off, and then it has branches and so on and so forth. So there's this really dense branching pattern, and the protein that is responsible for that is called ART23. Just in terms of its name, so ART stands for actin-related protein. So this is a protein that looks very much like an actin molecule. ART23 is a complex of two actin-related proteins. So ART23 is just two proteins that come together, and by itself, it doesn't really do much to drive actin assembly. Kind of like profilin by itself doesn't drive actin assembly. ART23 by itself doesn't drive actin assembly. But when it's in the context of other things, it does drive actin assembly just by creating more plus ends and more branching. It needs regulatory proteins, which we'll talk about in a bit, and actin filaments themselves. And let me just see. And the way this works, and here sometimes I really hate cartoons, I like this cartoon. So when we talked about the G-actin, we talked about how there's a smooth side and a cleft side. So in blue here, we have our ART23. And what you'll see is there's two proteins that are kind of drawn like actin. And in fact, as far as the cell is concerned, they have the same functionality as actin. So actin can assemble off of ART23. So when ART23 binds to the side of an actin filament, there's going to be another protein that comes in, turns it on. That's going to change the conformation of the ART23, and then when it does that, it can nucleate actin assembly, and you get a new actin filament. So if we go back here, at every branch point, there's going to be an ART23 that is in fact holding that branch onto the actin filament. Yeah, go ahead.
If you go back to the photo of the ART23, I'm just trying to understand like you said that the ARC2 and 3 are what look like the G-actin clefts themselves.
Yes, yeah.
What is the overall, like the larger blue structure there that is kind of anchoring it onto the...
Yeah, so I guess maybe to even explain that better, if you think about the plus end of an actin filament, there's going to be 2 G-actins there. And you've got your ARC2/3, you've got 2 things that look a lot like an actin filament plus end. And then that needs to be held on to another actin filament. And this large structure here is just kind of indicating that part of that molecule is actually binding to the actin filament. And that's what it's trying to show.
It's not a separate molecule that's just showing that it is bound to the...
Yeah. Okay. And that's like with cartoons, they're great until you push it too far, and then you're like, yeah, okay. So, all right. So what happens, I just walked through that, and this is kind of, close up here. And this is, just another sort of thing. So this is another electron micrograph, and we're really zoomed in here. So this sort of shadow here, this is going to be your ARC2/3. One thing that's really characteristic about ARC2/3 is that the branches make these 70-degree angles, and so if you were to go in and measure the angles, they would be about 70 degrees in each case. It drives activism. Yes, go ahead. The question is, why is it always 70 degrees? And I think it just has to do with the conformation of the proteins. So I don't think there's anything special about it. When it was first sort of discovered, when people were working out ARC2/3, it was kind of like, oh wow, it's always like 70 degrees. There must be something there that makes it always 70 degrees. So it was kind of a little observation that then also helped drive the discovery of ARC2/3, more than who cares about the 70 degrees. So, So, okay. All right. So we talked about these row family GTPases, and ARC2/3 is regulated by CDC42 through the protein WASP. And WASP stands for Wiskott-Aldrich syndrome protein, not the insect. So that's where the abbreviation comes from. And CDC42 stands for cell division control protein. And it happened, I don't recall if it has a molecular weight of 42 or what it was discovered, but the 42, very often when you see things like 42 in the name, it is the molecular weight of the protein. This is something that I should check and find out. So, but in any case, up here we've got our plasma membrane. It's got a lipid anchor. CDC42 is bound to the membrane. It's bound to GTP here. That is then activating WASP. So before we had this theme where Rho activated formins to modulate actin assembly. Here we have a case where CDC42, another Rho family GTPase, binds to a different protein, in this case WASP. It opens it up. And then there are also domains here that facilitate actin nucleation, and then that target this to the ARC2/3. And another sort of implication of this is new branches are going to be formed at the leading edge, right at the membrane, right? So this protein where ARC2/3 gets activated is right at the plasma membrane. And you can imagine with this treadmilling that actin is being assembled, new ARC2/3s are being added, branches are being formed, and it's like a treadmill. All of that stuff is going to move back, and then eventually cofilin is going to break it all down, create the G-actin, and then that will be recycled to drive the cycle again. All right. So to bring this all together, this is an incredibly famous summary describing the regulation of actin at the leading edge. And I'll just sort of walk through it. So I guess, yeah, okay. So just to put this in context, we've got our plasma membrane up here at the top, and there's going to be a receptor here in blue. There's going to be some sort of extracellular stimuli that's going to come in, and what that does is it's going to activate different Rho family GTPases and also PIP2. This is going to activate WASP, which also goes by the name SCAR, and I actually was really good friends with the guy that discovered SCAR. So he was working in dictyostelium at Emory, essentially the lab like right next to mine. We took classes together and stuff like that, and it got named SCAR. But in any case, so these proteins then activate ARP23 to initiate new filaments. So if we look over here, we have our actin filaments. The plus ends of the actin filaments are going to be right at the plasma membrane. The minus ends are going to be down here. The way that this is sort of color-coded in general, I'm not sure here. But in any case, as this actin assembles, it's also going to age. So we start off with ATP actin at the very tip. It's going to be hydrolyzed, so we have actin with ADP and phosphate. Those phosphates are going to pop out, and we're left with actin ADP, and then we have old actin back here. Just to kind of walk through your cycle, we've got profilin working at the tip to drive assembly. Cofilin is going to sever these filaments and cause them to create many minus ends. And then from these little fragments, the actin disassembles, and we have our ADP actin here. The ADP actin is going to bind to profilin. So profilin promotes actin filament assembly. Part of its job is it binds the G-actin and it opens it up, and it allows that ADP to be released in exchange for ATP. And then we have our pool of ATP actin. Once again, profilin, formins, different molecules acting at the surface are going to drive assembly. And these little dots here, these are going to be our ARP23s, and then those are going to form new branches. Through this process, the barbed ends, so the plus ends of the actin filament, they're going to be elongating, and they're going to actually be pushing against the plasma membrane. There's other forces we'll talk about in a bit, but as this whole thing moves backwards, our capping protein CAPZ is going to bind to the plus end of the actin filaments, and it's going to prevent assembly throughout the inside of the cell. So we just have assembly at the tip, and then it all sort of comes together. As you can see, at one level, there's a huge amount going on here, but everything fits together. Everything has a function, and I think it makes pretty good sense. But I'll just pause for a moment and ask if there's any questions about this figure. Yeah?
Just making sure, the small yellow triangles on the right side, those are all the ADF cofilin?
Yes,
Okay.
yes. Good point, yes. All right, anything else? Okay. And then one of the things that I think is really cool, so we just looked at that cartoon of actin assembly, and what's happening at the leading edge of this cell are these processes that we just looked at. And when you're looking at this movie, if you can simultaneously imagine all of this happening under the hood, that's the mechanics that's making these cells crawl. And then when we look at it in motion, it all sort of comes together and stuff like that. So there's stimuli, they're activating PIP2, different Rho family GTPases, it's activating profilin and formins, ARP23 is getting activated by WASP, or SCAR in this case, because, well, no, this would probably be WASP. And then eventually, the bacteria gets captured. So with that, that is the conclusion of today's lecture. Any questions?
And we saw this in the class before, but is there a reason why you don't just phosphorylate ADP to produce ATP? Why do they always have to exchange ADP for ATP?
Yeah, so the question is, why can't ATP or ADP be directly phosphorylated in the context? It actually, so in glycosylation, glycolysis rather, you do get substrate level phosphorylation of ADP to create ATP. So that does happen to some extent. It just turns out that in most sort of cells that are using oxygen to respire, about 90% of the ATP is being made in the mitochondria through oxidative phosphorylation. It's more just sort of a situation where overall, about 90% of the ATP inside the cell, about 90% is ATP and about 10% is ADP. So there's a much higher concentration of ATP than ADP. And more or less, the way that the cells are getting energy, quote unquote, through ATP is that because there's so much ATP around and a low concentration of ADP, when they let go of the ADP, there's about a 90% probability that it'll get replaced with an ATP. And that's kind of like what's going on. So, yeah, all right. So, other questions? Okay, all right. Well, in that case, I would say have an awesome day, and I'll see you later.